When we think about the human body, we think about its ability to heal. However, if the central nervous system gets damaged, such as a spinal cord injury, this damage is permanent. One way that research approaches this dilemma is by looking at embryonic development, since this is a period of life where all nerves are growing. A well-established model for neural development is the visual system. And in Dr. Eric Bergbauer's lab, we use CHIC as our developmental model. Additionally, we focus on a subset of neurons, retinal ganglion cells, which originate in the eye. They bundle together to form the optic nerve, and they continue on their path to the brain. These neurons have a defined path, which means that the neurons from the right eye will travel to the left brain, and the neurons from the left eye will travel to the right brain. They also organize in a highly specific manner. In our lab, we're interested in what molecules help guide these neurons to their destination. More specifically, while these neurons journey to their destination, they encounter many different molecules to help aid in this guiding process. So you can think about these different molecules as either being a stop signal or a go signal. One very important molecule, lysophosphatic acid, has many important biological functions. However, LPA has not been defined as having a role in guiding neurons to their destination. However, here at Winthrop, we're trying to be the first people to do this. Based on previous research, the evidence suggests that LPA is one of these stop molecules. So what I mean is, when a developing neuron interacts with LPA, you can actually see that it retracts or steer away. Therefore, my graduate thesis focused on a specific detector, which we refer to as a receptor, that interacts with LPA at the molecular level. Furthermore, my graduate thesis will investigate whether this specific receptor is responsible for helping communicate with LPA and to ensure that these neurons don't go to the wrong side of the brain. To accomplish this, I will use a new and exciting gene editing technology, CRISPR. This technology will allow me to make sure that this receptor can no longer function correctly, and therefore it cannot interact with LPA. Ultimately, the outcome of this research will provide us with a better understanding of neural development, but it also has the implications to be used therapeutically for nerve regeneration.